What's up, Tumna Lectures? In this video, we are going to be looking at what we call the liver lobule. If you find it helpful at the end of it all, please make sure you like the video, comment down in the comment section, and then subscribe. Let's go ahead and get started. In the previous videos, we have been looking at what we call the liver anatomy, the anterior view, the posterior view, the liver circulation. We talked that the liver is going to be having what we call a dual supply. And we said that when we talk of a dual supply, we are meaning that you're going to be having two kinds of the blood vessels which are going to be supplying the liver. We said that we also have what we call the hepatic artery proper that was entering the liver to supply what we call oxygenated blood, approximately 25% of the blood going to the liver. We also said that we also have what we call the portal vein, which was supplying the blood which was rich in the nutrients, even also what we call the pathogens, what we call the hormones, and even other kind of substances, alcohol and other toxins that was passing through what we call the portal vein, and all of it was going to what we call the liver. Okay. We also said that we also had what we call a drainage system of bile, which we called the bile, the common hepatic duct that was draining what we call the liver. So we said that that whole structure where all those kind of the tubes are passing, we called it what we call the porta hepatis. Okay. Now, we want to see what happens when this blood enters into what we call the liver. Now, the liver has what we call a structural functional unit, which we are calling the liver lobule. Okay. So now, when this blood enters here in this kind of the liver parenchyma, we are going to be having this kind of the liver lobule. Now, we have three types of liver lobule. The first one is what we call the classic liver lobule. The second one is what we call the portal lobule. And then the third one is what we call the acinus lobule. Now, in today's video, we are going to be focusing on what we call the classic. Okay? So now this one you are seeing is what we call the classic. Is what we call the classic liver lobule. Okay? This is one which is going to be so much common. Okay? Now, in the liver parenchyma, we're going to be having this hexagonal structure, which we are calling the classic liver lobule. Now, when this kind of the portal vein enters to the liver, it is going to be giving off what we call tributaries, or what we call the venues. Okay. Even also this hepatic artery proper is also going to be giving off tributaries, which we call the arterioles. So now, this hexagonal structure is going to be having 6 sides on it. So now, on every corner, we're going to be having a tributary from the portal vein, a tributary from the artery or hepatic artery proper, and even also we're going to be having a branch of what we call the biliary system, which we call the bile duct. Okay, so now on every corner you're going to be having those kind of 3 tubes collectively as what we call the portal triad. Okay, so now having seen all that, let us now cut on the whole of this classic liver lobule. We zoom in outside here and see exactly what is going to be happening. Okay. Now, this one here is going to be what we call our classic liver lobule. We want to see exactly how the blood is going to be passing through this kind of what we call the liver lobule. But before that, we need to first understand these corners. What are some of these structures which are going to be on all of these corners? We need to first understand how we call all those tubes. Now, this kind of the red tube, this red one here, is going to be a tributary of what we call hepatic artery proper. So now this one is going to be a tiny arteriole from hepatic artery proper. So now this one here is going to be what we call hepatic, is what we call the hepatic arteriole. Okay, it's going to be what we call hepatic arteriole, meaning that it was coming from what we call hepatic artery proper. And then this one here is going to be now a tributary from the portal vein. So we can just simply call it the portal. We can simply call it the portal vein, meaning that is draining blood from the hepatic portal vein into this kind of tributary, which we are calling the portal vein. Then we also have this structure which is draining what we call the bile. And this structure here is what we call the bile, duct. Okay. So now these structures are going to be the same on all corners of this hexagonal structure we are calling the liver lobule. Now, these 3 structures collectively are what we call the portal triad. Okay, these structure 3 are what we call the portal triad. Okay. However, you should put in mind that I will also have some other tiny structures that are going to be entering through this portal tract. We also have some other structures. For example, we have some structures like the lymphatic vessels. Okay, we also have some other structures like the lymphatic, what we call the lymphatic vessels. Okay, lymphatic vessels that are going to be draining excess of the extracellular fluid, okay, into what we call the celiac lymph nodes, okay. And then also should not forget that you're also going to be having a certain kind of the hepatic plexus, okay. So you're going to be also having what we call hepatic plexus, which is going to be more of what we call the innervations to what we call the liver. And of course, you're going to be having the sympathetic fibers coming from what we call the celiac ganglion, and even also parasympathetic fibers coming off with what we call the vagus nerve. Okay, so now they are such beautiful things. Okay, now I want to look at when the blood in this kind of the hepatic arterial, and then also this kind of the of what we call the portal vein. Now, when it enters into this tube, so now how do we call the whole of this tube? This tube is going to be a capillary, okay? And this kind of the capillary is specific, super very important. You know how this capillary is called? This capillary here is what we call the sinusoid capillaries. Okay, so now this one here is what we call the sinusoid capillary. Okay, is what we call the sinusoid capillary. So now blood from what we call the portal vein and then hepatic arterioles are all going to be draining in this kind of the sinusoid capillary. It means here blood is going to be mixing. Okay, you're going to be having mixing of what we call the blood. It is such another beautiful thing that the blood which is rich in nutrients and the blood which is rich in oxygen is now meeting in this kind of what we are calling the sinusoid. Now, the sinusoid is going to be lined by these endothelial cells. You see these endothelial cells? Super very important. These endothelial cells are going to be having what we call fenestrations. They're going to be having a lot of these big gaps. You see these big gaps that are going to be allowing all the the nutrients to pass from here, and then they enter into this space. You know, from what we call the sinusoid capillaries, before we reach these cells which are like in the plate, we have this space here. Okay, you see this space here, this space here, this space here, this space here. In other words, from the sinusoid capillaries, we have another important space which is going to be allowing for what we call exchange of what we call nutrients before they access what we call the hepatocytes. How we call this this space here. This space here is what we call the space of DC. Okay, is what we call the space of DC, but some people can call it the perisinusoidal space. This space is super very important. It is going to be one of the exchange area where it's going to be having the absorption of these the substances into what we call the hepatocytes. Okay. So now I also have these cells which are arranged in like a plate. You know how these cells are called? These cells here are what we call the hepatocytes. Okay, these cells here are what we call the hepatocytes. And these hepatocyte cells are going to be arranged in a plate form. Okay, they are going to be arranged in a plate form. All the functions of the liver are going to be taking place in these the cells we are calling the hepatocytes. Super very important. Okay. Then within the space of DC, we have this cell. This cell is super very important. We have these cells in the space of DC. These cells are super very important. This cell here, which is going to be in what we call the space of DC, this cell is what we call the ito cell. These ito cells are super very important in what we call storage of what we call vitamin A. Super very important in storage of what we call vitamin A. Okay, these ito cells are super very important in storage of what we call vitamin A, and even also during any of the liver injury. Whenever the liver gets injured, the inflammatory mediators are going to be stimulating these the ito cells to release a certain of what we call the collagens. And for a lot of the collagen piles in this space of DC, is going to be now increasing what we call resistance to the flow of what we call the blood in what we call the sinusoids. And in this way, the liver is going to be start to be fibrotic. And of course, when the liver becomes fibrotic, there's going to be a resistance in blood flow to what we call the sinusoid. So now what happens is that the blood is now going to be backing up into what we call the portal vein. And in that way, if you have the backup pressure into the portal vein, this will lead into what we call the portal hypertension. Okay, so it's such super very dangerous situation. Okay, so now when you come again into what we call the sinusoid capillaries, we also have this macrophage. This super very important in the process of phagocytosis. You know how these phagocytes are called? These cells here, this one here, this one, they're going to be residing within the sinusoids. This one here is what we call the Kupffer, the Kupffer cell. This one is a super very important, playing a role of immunity. You know, when you have a lot of the substances being absorbed, like for example, may absorb the pathogens, okay, the toxins. So now this the Kupffer cell is going to be making sure that all the pathogens are phagocytosed down, such that before this blood leaves the liver to go into the general circulation, it is clean and free from infections. And that's why the liver also plays a role of what we call the immunity. Besides that, this the Kupffer cell is going to be also breaking out the worn out red blood cells. And the final product is going to be what we call the unconjugated bilirubin, which is going to be entering what we call the hepatocytes for the process of conjugation before the bilirubin gets stored in form of bile. It is such another beautiful thing. Okay, so now the Kupffer cells are going to be residing within these capillaries for immunity to phagocytose any kind of pathogens that has been absorbed with the food. Then we also have these green tubes. These green tubes are super very important. You know how these green tubes are called? These green tubes are going to be draining one of the most important substances, which we are calling the bile. So now, for that reason, these green tubes here, they are simply called as what we call the bile, canaliculi. Okay, is what we call the bile canaliculi. That's going to be draining what we call the bile. You know, now we're going to be having blood from the venule and the artery moving towards the center, but you're going to be having now the bile canaliculi moving the bile in opposite direction. Okay, so now this is how something becomes very much beautiful. Blood is moving centrally as the bile is moving in the opposite direction. But now blood is moving to this center kind of the tube. You know how this tube is called? This tube here, which is going to be at the center, is going to be a vein, and it's just simply called the central vein. Okay, so now this tube here at the center, this one here is what we call the central vein. Okay, so now after all the blood has been processed and it has a lot of the nutrients that now has to go into the general circulation, it has to come to this what we are calling the central vein. Now the central vein from other lobules are going to join together, and then they form what we call the interlobular vein. And then multiple interlobular veins are going to be coming together, they form what we call hepatic vein. And then hepatic veins are going to be now draining direct into what we call the inferior vena cava, and then the blood goes into what we call the right atrium. Okay, it is also such another beautiful thing. So now the central vein, for the case of what we call the classical liver lobule, is going to be super much in what we call in the center. Okay, in this this hexagonal arrangement. Okay, now having seen all that, I want to pay much attention that these hepatocytes which are going to be in a plate form, I want to first extract two of them, and we see that one face of these hepatocytes is going to be facing what we call the space of DC. Again, we're also going to be having another face which is going to be facing what we call the biocanalicular. What am I trying to mean exactly? So now, if this one is my hepatocytes, okay? If this one is my hepatocytes, okay? So we are going to be having one face which is going to be having these the tiny extensions which we call the microvilli. This one is going to be now facing what we call the space of DC, where the nutrients are going to be coming into contact before they're transported into the liver cells for the process of for other metabolic processes to take place. But again, on its opposite side, you're going to be having now, you're going to be having what we call the bile system. Okay, so now you're going to be seeing that the basolateral side of this kind of hepatocytes is going to be facing the space of DC, where the nutrients are coming from, and then the apical side is going to be facing what we call the bile system. It is such another beautiful thing. So now, in between these cells here, you're going to be having some kind of the tiny bile canaliculi. Okay, so now these kind of the bile canaliculi are going to be residing inside of these cells. Okay, and these cells are going to be held together by what we call the tight junctions. That is something which is super very important. The cells, all the sides are going to be performing different processes. It is such another beautiful thing. Okay, so now once the nutrients from what we call the sinusoid capillaries leaves through these kind of fenestrations into the space of DC. Now, these hepatocytes are designed with different transporters, okay? Different transporters of different substances. Now, we are going to be seeing now the nutrients, what we call the unconjugated bilirubin, getting transported into what we call the liver cells, which are calling the hepatocytes, for different metabolic processes. So now, let's go ahead and see what are some of those processes that are going to be carried out by what we call hepatocytes. And by doing that, we are going to be now explaining what we call the functions of the liver, because all the functions of the liver are going to be taking place in this kind of liver lobule, particularly to what we call the hepatocytes. Okay, so let's go ahead and see the functions of what we call the liver. Okay.